We in the FSHD Europe organize these webinars, and that is because we want to increase knowledge about FSHD. We collaborate with clinicians and researchers from neuromuscular centers in European countries, and they are all a part of the FSHD European Trial Network. And now the important guest of the night, our FSHD Europe scientific advisor, Julie Dumonceaux. She's a professor of translational myology at the University College in London. She leads a research group working on neuromuscular diseases, which is strong focus on FSHD. She will tell us about her work and brings together patient samples, disease models, molecular mechanisms, biomarkers, and therapeutic development, including gene therapy approaches. And now she will present how biobanking can support research in rare neuromuscular diseases, from understanding disease mechanism to improving clinical trial readiness. So warm welcome to you, Julie, and I lead the floor to you.
Thank you very much, Caroline. I'm going to share the screen. Can you see the screen?
Yes, we can see the screen.
Okay, so let's start. So I would like to just to start by saying that I'm a researcher. I do not manage a biobank. I do not work in a biobank. So my talk today will not be a description of what a biobank is exactly, even if I will mention it. But today I would like to really focus on practical questions. Why researchers sometimes ask for samples, and how biobank may help research in FSHD in particular. So the presentation will be divided in 5 parts. First, briefly what a biobank is. Second, why it is important for FSHD. Then we're going to look at the different types of samples that can be collected and what can be done with them. We will discuss some concrete examples of biobank use in clinical research. And finally, we will conclude with an overview of ethical issues and data protection. So let's start with what is a biobank. So I'm sorry, that's a very, very busy slide. But the goal of the slide is to give an overview of what a biobank is compared to a research lab. It is very important because actually both biobank and research lab share common points, but they also differ in major points too. The most important common point is the fact that patient samples can be found in the two structures, but their goals are different. Put simply, the biobank collects the samples and the laboratory use them for specific purposes. So the biobank on the left-hand side builds a long-term resource. It carefully collects and stores the samples. But a biobank is not just a giant freezer. It's actually a highly sophisticated system designed to ensure that each samples remain in optimal condition. It does not necessarily have a predefined goal in mind, other than to help researchers and clinicians to better understand disease, to develop new treatment. The samples are collected actually for future use. So the biobank acts as a custodian and manages ethical consent forms. The biobank also collects some patients' data that may vary from one study to another, but they frequently include the date of birth, the sex, the disease, the genetic data, and etc. On the other hand, on the right-hand side, the research lab is like the biobank user. It requests samples to the biobank to answer a specific scientific questions. Usually, the lab sends a summary of what it would like to do and how many samples it needs to do so. And the biobanks verifies that the samples requested by the lab can indeed be used for the research the laboratory intend to conduct. For example, the biobank will verify that it has the right to share certain health data with the lab. So basically, the lab works under the biobank's rules. But sometimes a research lab can also collect samples and have a research collection. That usually means samples and data are collected for a very specific study to answer a specific research questions. So a biobank is different, is a more structured and usually more long-term. The aim is not only to answer one specific question, but to store samples safely, to keep traceability, and to make the samples available for future approved research under clear governance. But today, I'm going to use the word biobank in a broad sense because whether it is a biobank or research collection, the central question is still the same. Why can it be important for patients to contribute samples for research? And why do we do with these samples? We, I mean researchers and clinicians. Okay, so let's start with the samples and what you may be able to choose for your samples and what information can be used for. So the samples can be used for different purposes, but that's the patient who authorizes for which purpose. And here you have some examples. You can choose to have the samples used, for example, for one particular study only. You can authorize the storage of the samples for future research, but importantly, when you say yes to storage, it does not automatically mean that you agree to every future work. Importantly, samples can be linked to health information such as diagnosis, clinical score, MRI data, and actually that's this linkage that is very important because it often makes the samples clinically useful because we need to have an integrated view of what is happening in the patient. Samples are de-identified before being shared with researchers, and not everyone can access the samples, but access should be reviewed, approved, and controlled. A patient, I'm gonna, here, for example, a patient may agree or not to be contacted again for future studies, updates, or additional consent. And here, finally, a biobank is mainly for research, and researchers usually study de-identified samples and data. So, importantly, individual research results are not usually returned. However, in some occasions, participants may receive instead the general information about the study findings. So, the second point I would like to discuss tonight is why is this infrastructure vital for FSHD? So to answer this question, it's very important to have in mind that FSHD is a rare disease affecting 1 in 800 to 1 in 12 and in 8,000 to 12,000 people worldwide. And because it's a rare disease, it means that there is a very limited access to biological material. And that's the reason why every donation is really priceless. And researchers may need FSHD samples because actually the clinical observation alone is not enough. A medical examination can reveal the consequences of the disease, but not the underlying biological complexity, which is not visible. But access to patient samples allow us to connect the patient's experience, health data, such as strength or MRI scan, and the FSHD biology. And this is how researchers were, for example, able to scientifically document the patient's feeling about how the disease progressed. For example, patients often describe the disease as progressing slowly, but what does it mean scientifically talking? So when we use MRI-guided biopsy, after using them, researchers were able to see that some muscles area were actually deteriorating while some others were not moving. And this was a very important information to better understand the disease progress. So by using these samples, researchers can study the dog's form biology, how to switch it off, why symptoms and severity differ between patients, and of course, they can also develop treatment. And here you have a concrete example of what we can do in the lab using patient samples. And here are pictures taken in my own lab. For example, we may have access, and here we had access to small muscle biopsies. These muscle biopsies were cut into very, very small pieces using, as you can see here, forceps, scalpels, or scissors. And these very tiny muscle pieces were then placed here into what we call culture plates, like the one you can see here. So the small pieces of tissue stick to the plastic surface on the plate, and then we add liquid on the top called culture medium, which contains the nutrients the cells need to survive and grow. And little by, here you have the small pieces, and little by little, the cell starts to migrate out from the small biopsy pieces. Here you can see them. That's one cell, another cell, etc. And they colonize the plate. And then we can collect these cells and sort them. So when we keep only the muscle cells. And this is what you can see here, because we can check that we have selected the muscle cells by staining them with the muscle marker, which is here visible in green. And in blue, you have it the nucleus. And you can see, for example, that this nucleus is not a muscle nucleus because it is not surrounded by this green marker. And this is not surprising because actually, when we take a muscle biopsy, we collect the muscle tissue, of course, but also some other tissue that are important for supporting the muscle, such as the blood vessels and connective tissue. So then we have a mix of different cells in culture. Then the muscle cells that are called myoblasts can fuse with each other. And when they fuse here, they form what we call a myotube. And in a petri dish, these myotubes are the simplified version of the muscle fibers. And here you can see one researcher in my lab, Virginie, looking under the microscopes at cells that came from patient biopsy. And here you have a video. You have a video of Solène in my lab too, when she retrieves cells to culture them. So here you have the cells. You have the cells are stored in what you saw, these enormous tanks that are very, very cold because the cells are stored in liquid nitrogen at minus 185 degrees Celsius. And here you have a cryo vial where the cells are. And Solène is wearing effective protective equipment to protect her from the liquid nitrogen And so she's wearing face shields, apron, and particular gloves. And the access to this kind of room is totally restricted, as well as the access to the tank. That's an example of where the cells are stored. So what exactly can be collected? Actually, there is a whole spectrum of samples that can be collected. And the simplest is saliva, which is obviously very easy to collect and which give access to DNA. Urine can also be collected. Then we have blood. And from the blood samples, we can isolate different things. We can isolate the serum, the plasma, we can isolate the DNA too. And sometimes we can even conduct experiments using the blood cells, the cells that are in the blood directly. And blood is very interesting because it's minimally invasive, it's quick, not expensive to collect, and we can do blood withdrawal multiple times. So it's very useful to make a longitudinal assessment in patient. And finally, we have the muscle biopsy, which is a more complex and invasive procedure. And a biopsy is never done just in case or by chance. A biopsy is only requested when there is a real underlying scientific question which requires a direct access to the tissue. What is very important to know is that saliva, blood, or muscle cells don't tell the same story. They provide really different information. So for saliva, for instance, for example, is used to confirm genetics. It can help study to understand why there is the severity differs between patients. We can look, for instance, whether patients with the same genetic default have the same or not symptoms. The blood reflects different things because it reflects the signal sent by all the muscles, the entire musculature in the body. So these are called systemic signals. And the blood strongly supports biomarker research. And several biomarkers have been proposed by different research teams. These markers reflect different states of the muscles. And as the muscles deteriorate, it will gradually change physiologically. And this will be partly reflected in what is found in the blood. So in the blood, we can find markers of DOX4 expression, markers of inflammation, markers of good muscle health, or even we can look at if the muscle fiber that are breaking down. And the idea is to use these markers as marker of disease severity, for instance. But it's important to keep in mind that sometimes it's difficult to use this marker because they are not necessarily only linked to FSHD. Some of these markers can vary after eating or after exercise or after being infected by a virus like the flu. So therefore, we need to be extremely cautious when we interpret the data. Another use of the biomarkers is to determine if a therapy is working, as certain markers may change with efficient treatments. These markers are markers of therapeutic efficacy or target engagement. Another possible use of biomarkers is to be able to predict the progression of the disease, for instance, to be able to say if a patient has an increased risk of losing the ability to walk in the next 3 years, for instance. But for the moment, such a biomarker does not exist for FSHD. Finally, the muscle. So The muscle biopsies allows us to look at the state of the muscle at a given time and place. It's really highly precise, but at the same time, also very reductive because it doesn't represent what's happening throughout the entire body. It's like comparing a photo to a film. A film gives the overall view, but the photo is taken at one specific moment. So the photo provides a snapshot, and there is no guarantee that it's the same photo elsewhere. If you take the photo a little bit later, there is no guarantee that it will be the same photo than earlier. And actually, in fact, we can be sure that it won't be the same photo. But it's a snapshot, and a very useful snapshot because it will give us access to very useful information. We can look at the structural changes in the muscles that can be linked to MRI to better understand what is happening biologically in patients, for instance. We can look at the mechanism of the disease, and the muscle biopsy can also be used to identify possible biomarkers. So it is very important to have all this information, and it's when we can fit all the pieces of this puzzle together that we can get the best integrated view of the disease. So now, concretely, how does all your information fit together to accelerate research? So let's take the example of the RESOLVE study, which is a large international study on FSHD, started recruiting patients in 2018. The idea in this particular study was quite simple. If we want to be sure that we can assess treatment efficacy, we first need to know what we should measure. Do we need an MRI? Do we need a blood sample? Do we need to perform functional testing? What is the best one? And in FSHD, it is not a trivial question because the disease progresses slowly and not always the same way from one person to another. So RESOLVE was the first big study for trial readiness. Patients were followed over time. They had clinical assessment, MRI scans, functional tests, and blood samples. And by assembling the information together, the goal was to identify reliable ways to measure disease changes and to validate specific clinical assessment. But all in all, the aim is very practical, actually, is to make future clinical trials better designed, more sensitive, and hopefully more efficient. Another study is the MOVE Plus, which is also a trial readiness study. But the goal this time was to identify meaningful outcome measure. So this can be used in future clinical trial, and it can be used also to better understand disease progression. So MOVE was designed to answer specific questions such as which aspect of the disease change over time? Can we measure that in the blood? Can we measure that in MRI? So that's why biobank were very important in this kind of study. Which method are the most sensitive to detect progression? Do we have to perform muscle biopsy, or can we use surrogate endpoints using a biomarker in the blood? By using the data on these clinical trials, we can better understand the disease and we can have better outcome measure for future clinical trials. The MOVE Plus takes one step further because it combines the clinical information with the whole body MRI, the biological samples, and for some patients, a muscle biopsy. And the idea is to connect the patient's experience with imaging and biology to identify biomarkers that could improve future trials. And now I would like to jump on the last section but which is one of the most important pillars of the entire system, the ethics and security. It is very important because a biobank cannot exist without the complete trust from the patients. And as we said before, the ethical framework is absolutely strict. And when we talk about ethics, we are really asking a simple question. Are samples being used in a way that respects the wishes, privacy, and interest of the people who donated them? So as soon as the sample arrived, the data is immediately coded. Personal identity is ultra protected and never disclosed to the lab. Access to the resource is also very limited to authorized researchers only, and protecting the patient contribution is an absolute priority. However, you may have also seen in the news that there was a recent incident involving the UK Biobank. What happened is that some participants' data ended up online, and this is not something to downplay, even though the name were removed and from the health data, it's obviously remains sensitive. However, these kind of events are rare, and patient participation, including yours, is still absolutely essential so that we researchers and clinicians can continue our work, namely understanding how FSHD works and finding treatment for everyone. And finally, this map shows some of the major biobanks and sample collection relevant to neuromuscular diseases across Europe. These biobank differs in size, organization, and focus, but they share the same goal to preserve patient samples and make them available for approved research projects. Importantly, no single center has enough patient samples to answer every research question. So the collection of samples across many countries is essential. And researchers are not limited to the biobank of their own country. They may apply to access samples from different biobanks depending on their needs. Biobanks do not work in isolation, and researchers are often collaborating across countries. But if your thoughts are now, can I contact a biobank directly? I would say that sometimes, yes, you can, because for some biobanks, you have a public website and contact details. However, the usual route is through your clinical team, the patient organization, or research study. And in most of the cases, patients do not donate samples directly to a biobank. They are usually collected through hospital visits, natural history study, and sometimes a clinical trial. So you don't need to wait for a clinical trial to contribute to research. So finally, I would like to thank you. I would like to thank, of course, all patients and family who support FSHD research, for those who contribute to samples, helping us study the biology of FSHD, and for taking part to this webinar. I'm happy to take your questions now.
Thank you, Julie. Do we have any questions? I have one. If there is no biobank in your country, can you ask your neurologist to send it to someone else?
That's a question I cannot answer, but I guess that you can, because your neurologist can collect the samples and then he can address the samples to the correct biobank. Yes, I would say yes. But I would like to precise that I'm a researcher. I'm not a medical doctor, so I don't see patients.
And Ria?
Thank you, Julie, for this nice presentation and overview. I was wondering, is there a feedback back to the patients that they know what the biobanking results, what the result was, or what happened with their samples, or what kind of research, or do you just give the consent for any research and you don't know what happened with it? Is there any feedback to the patients about that?
Usually there is no feedback to the patient specifically, but sometimes the patient can have a general feedback on the finding. It depends on the biobank and on the research proposal.
Do we have any more questions? I guess everything was clear then. A good job, Julie. Thank you so much.
Thank you very much.
So I will leave you with just to let you know that don't forget that it's World FSHD Day on Saturday. So share all these posters that are on Facebook and Instagram and all, so we can make the world aware of us. So thank you so much for tonight and have a good night.
Thank you.
Thank you.
Bye.
Bye-bye.
Thank you. Bye-bye.
Thank you.